We found so many things that were really pretty unexpected for us. I mean, one of the biggest things that we were surprised by was how completely normal at the level of gene families the octopus genome looked. So when we sequenced it, we kind of went through and looked for different families of genes. And when we did that, we found that they have kind of the normal number and kinds of genes that are found in other animals like snails and worms. And this was kind of particularly surprising to us because cephalopods have very big genomes. So the genome of the species of octopus that we sequenced, Octopus bimaculoides, is about 2.7 billion base pairs. Wow. And that's about 90% of the size of the human genome, which is about 3.2 billion. And this is many times bigger than the genomes of other animals. So we thought that they'd actually gotten this big by making multiple copies of their genomes. And this may sound like kind of a crazy idea, but this is actually what happened along the lineage of animals that led to vertebrates, like mammals and fish. So mammals and fish actually have multiple copies of their genome, multiple copies of each gene. So when we were looking through the octopus genome, we didn't actually see that signal, and we were really surprised by that.
So do you have an idea what all those all those genes are used for?
Some of them, yes. Some of them, no. So we found about 10% of the genes didn't have a match to anything in any other animals. So that's really exciting. And we know that there's a family of these genes that are called the reflectins, and they're involved in the coloration, the structural iridescence that's formed in the adaptive camouflage response that these animals have. And, so this is just one gene family out of a whole bunch. So there's a lot to discover here. We also found that there were a couple of gene families that were massively expanded. So most of the gene families just had a single copy of the gene, of the different members. But there was this one family that's called the protocoadherins, and this is a group of genes that are involved in cell adhesion, and they've been studied in vertebrates where they play a role in setting up neuronal wiring. But they hadn't been really described in other animals, and so we thought we were pretty special. We've got about 60 of them. So we were really shocked when we looked in the octopus genome and found 168 of them. And near as we can tell, the last common ancestor of vertebrates and octopus only had one copy of them, of this gene. So, they had to expand them independently. And I think, things like that are really exciting.